Welcome back to part 2 of our basic pathology video review of immunity, some of the basic players in immunity, some of the hypersensitivities, and we're going to get into amyloidosis. So stay tuned. So if you remember in previous videos, we talked about the major histopathology complex. We talked about class 1 and class 2. And remember that class 1 is displayed by all nucleated cells. So is this going to include red blood cells? No, of course not, because they do not have a nucleus. Anyway, we'll talk about how natural killer cells would be activated or not activated and why this happens. So remember that the healthy cells express self-class 1 MHC molecules, which are recognized by the inhibitory receptors, and this ensures that the NK cells do not attack the normal cells. So if you were looking at an NK cell and it was coming into contact with a normal cell, you know that normal cell would be displaying this self-class 1 MHC self-peptide complex, and the NK cell would then have these receptors. They either have an activating receptor which would destroy the cell, or an inhibitory receptor. So in this case, the inhibitory receptor would connect with the self-class 1 MHC self-peptide complex, and then the NK cell would not be activated, and there would be no cell killing. So this is the normal situation. It's like having a password. The normal cell will present the password, and then they would not be killed. Then you think of the situation where the NK cell is activated and there is killing of the infected cell. So in what case would this happen? So how does this process work where the inhibitory receptor is not engaged, but the activating receptor is engaged? And activating means that the NK cell would attack the cell. So this, of course, is not a normal cell. Something's going wrong in that the inhibitory receptor is not engaged. So the cell has been infected or stressed, and the class 1 MHC expression is reduced. So the inhibitory receptors are not engaged, and the ligands for the activating receptors are expressed. Activating again meaning that it's going to get killed. So the result here is that the NK cells are activated and the infected cells are killed. So how does this happen? So you imagine the NK cell, natural killer cell, comes into contact with this troubled cell, and this time instead of the inhibitory receptor being expressed, is not expressed because there's a problem. Instead, there is an activated ligand for the NK cell, and this means that it's going to be killed. And so the result here is the killing of the infected cell. So remember that the virus, for example, would inhibit the class 1 MHC expression, and thus this would be lead to an increase in the expression of the activating ligands for the NK cell. So this is not a good thing. Okay, so in continuing on, we're going to talk a little bit more about the Mhc pathway for class 1 and class 2. Remember that there is antigen processing, and they are displayed by the Mhc. And to start off with, I'm just going to ask a question. So concerning class 1 MHC, what T cells are these going to be involved with? So these are CD8. And how about for the class 2 MHC pathway? What are these going to be interacting with? What are they going to be, where is this antigen going to be presented to? Of course, these are to the CD4 cells. So let's talk a bit more in detail about how this antigen is taken in and presented. So just as an overview, for the class 1 Mhc pathway, peptides are produced from proteins in the cytosol and transported to the endoplasmic reticulum, where they bind to class 1 Mhc molecules, and then the peptide Mhc complexes are transported to the cell surface and displayed for recognition by CD8 cells. So this is how it happens for class 1 MHCs, in the class 1 MHC pathway. So what happens, just as a review, is that the cytosolic protein is internalized into the ER first, and then it binds to the cell receptor and to the CD8 cell. So that is how that happens. But how about for the class 2 pathway? In this case, the proteins are ingested into vesicles and degraded into peptides, which then bind to the class 2 MHC molecules, and they're not transported in the same vesicles. And then these the peptide complexes are expressed on the cell surface and recognized by CD4 cells. So it's a little bit different here. Remember here that we have endocytosis of the extracellular microbes actually brought in, and it's brought into an endocytic vesicle, and then it is transported and then presented to the CD4. So it's not going through the ER, whereas in the class 1 situation, where they're found in where the peptides are produced from proteins in the cytosol, they're transported first to the ER, where then they bind to the class 1 MHC molecules, and they're presented to the CD8 cell. So that's just a very quick overview. Good to know that, I think. So that was just a very quick review of the immune system, the innate and acquired systems. And now we're going to talk about the immune system when things go wrong, the hypersensitivity reactions 1 through 4, some of the autoimmune diseases, and we'll talk about primary or genetic and secondary or acquired immune deficiency syndromes, also known as IDS. So here we're talking about inflammation, which is not due to external pathogens. So let's talk about these. First, we'll talk about some of the hypersensitivity reactions. So remember the different type of reactions. We have 1, 2, 3, and 4. So I will name the type of reaction. You will name the hypersensitivity reaction. So how about if we have an immune complex mediated hypersensitivity? So remember, this is an antigen and an antibody, and they are moving around causing trouble. That would be hypersensitivity type 3. How about if we have cell-mediated hypersensitivity? And this is a delayed reaction. This, of course, is type 4. How about immediate hypersensitivity? Seconds to minutes. This is type 1. And then finally, we have antibody-mediated hypersensitivity, which is a hypersensitivity to self-antigens, often seen in blood transfusions. So that is type 2. So there we go. So let's get into it a little bit more. And we're going to start talking about type 1 immediate hypersensitivity. So what do we know about hypersensitivity? So what could this lead to? Of course, anaphylaxis, shock, edema, dyspnea, death. And so with an allergen exposure during the immediate phase, we have what type of degranulation from what type of cells? Of course, these are from mast cells. I believe basophils are also involved, but when we talked about before about IgE being released, those of course from the mast cells. We have vasodilatation, vascular leakage, smooth muscle, bronchospasm, and of course we have the release of histamine. And in the late phase, hours and days, we would have eosinophils, PMNs, and T cells. So everyone's in here. Everyone's got something to do in this immediate hypersensitivity type 1. How about for type 2 hypersensitivity? Remember what type of hypersensitivity that is? Of course, remember that is the type 2 antibody-mediated immunity. All kinds of problems happening here. Later on, we'll discuss 2 big examples that are often discussed. For example, Myasthenia Gravis and Graves' disease, which often come up anyway. Let's just basically have an overview. Remember that these antibodies are attaching to cell surfaces, and there is opsonization. So which would the antibodies would are then they're uh, coating the surface and preparing them for phagocytosis. So this is a big problem. So we have signaling proteins, we have complement fixation, and we have a cascade of complement. And you can remember what that major one is, the one we hear coming up often, and that's C3. And there are many different types of the C3, if I remember correctly. But we have C1Q, C1R, C1S, C2, C3, C4, and C5, all leading to inflammation. So complement fixation leading to inflammation. And then with this, we have lysis. So this is leading to destruction of the cells by rupturing or breaking the cell membrane. So this is the result of this antibody mediated immunity. All kinds of problems happening here. Major ones that I want to talk about, we have two ways. First one, opsonization and phagocytosis, where there is an opsonized cell that's been coated. Then we have the complement activation involving C3b, and with the Fc receptor on the phagocyte, we have a phagocytosed cell, and we have the ending up in phagocytosis, whereas the second part, we have complement and Fc receptor mediated inflammation. We have what we have is a resulting inflammation and tissue injury. So different from type A, where we have opsonization and phagocytosis resulting in the phagocytosed cell. Here we actually have a release of histamine, for example, inflammation and tissue injury, neutrophil enzymes, reactive oxygen intermediates. Anyway, what's happening here is that we have the Fc receptor and we have complement activation, and then complement byproducts such as C5a and C3a. So just to summarize there, in A, we have opsonization of cells by antibodies and complement components and ingestion by phagocytes. But in B, we have inflammation induced by antibody binding to Fc receptors and leukocytes and by complement breakdown products. So quite a different result here in this antibody-mediated type 2 hypersensitivity. So let's talk about the type 2 diseases. Can you name any of these diseases? So to name a few, we have autoimmune hemolytic anemia, or AHA, idiopathic thrombocytopenic purpura, or ITP, Goodpasture syndrome, which is affecting the basement membrane of the lung or the kidney, so this resulting in nephritis and lung hemorrhage. And we have the often talked about rheumatic fever, which results from a strep infection in a child, leading to trouble with the heart, joints, skin, and brain. So a very often talked about one there. Then we have myasthenia gravis, which results in the neuromuscular junction. If you remember how that works, we have a blocking here where the antibody is causing, well, what it's doing is it's inhibiting the binding of the neurotransmitter of acetylcholine to the receptor, and which results in disturbing the normal function of the receptor. So we'll talk about that in a second anyway. How about when you have trouble with the TSH receptor? What is that disease called, this type 2 disease? Of course, that's Graves' disease. How about when you have the gastric parietal cells affected? Then what would this be? This, of course, is pernicious anemia. So let's talk about myasthenia gravis. What happens here? Of course, this is antibody-mediated cellular dysfunction. What's happening here is the antibody is attaching to the ACH receptor, the acetylcholine receptor, and it is then inhibiting the binding of the neurotransmitter to the receptor. So what does this do? What's happening here is that it impairs the neuromuscular transmission in myasthenia gravis, meaning that it's similar to someone pushing on the doorbell and keep on pushing it on it, and you'd expect that it would block it, but actually what's happening is when I'm getting confused here, I'm talking about Graves' disease when I should be talking about myasthenia gravis. Here what's happening is the anti-receptor antibodies disturb the normal function of the receptors, and the antibodies to the acetylcholine receptor impair neuromuscular transmission in myasthenia gravis, which leads to progressive muscle weakening. So this is quite easy to understand because of a blockage here. So remember here that there is no contraction as a result in myasthenia gravis because the antibody complex is I get confused here. Let me just double check this. Wait, did I just say antibody complex? I'm getting confused here. I mean, this is good for you to watch this video so you can correct me. Anyway, I'm correcting myself here. It's not an antibody complex that we'll be talking about. Type 3, that would be the immune complexes. We're talking about the antibodies here that are getting in the way of the receptors here for myasthenia gravis. And this is causing the inhibition of the binding of the neurotransmitter to the receptor. So how about for Graves' disease? This is the opposite. Okay, in this case you can't turn it off. What's happening here is that the antibodies against the thyroid stimulating hormone, or TSH, receptor activate thyroid cells. So they're actually blocking the receptor. But what's happening is that the antibody is stimulating the receptor even without the hormone. So then this results in Graves' disease. Okay, so now let's move on. We're going to talk about immune complex mediated hypersensitivity. Actually, let's take a break here and we'll move on to the next video.